Welcome back, guys. Today we are going to discuss an important topic of cell membrane in the cellular physiology modules. So in this cell membrane, the first we are going to discuss is fluid mosaic model. I hope everyone is familiar with this model. This fluid mosaic model was proposed by Singer and Nicholson. Singer and Nicholson proposed this fluid mosaic model. This model suggests that there are three important macromolecules in cell membrane. There are three important macromolecules in cell membrane according to this model. First is proteins. Proteins are the major ones, constituting nearly 55% of the total. Then comes the lipids. They contribute to nearly 40% of total. And the least important is the carbohydrates. Carbohydrates are least important. They contribute to nearly 5% of total. So these three important macromolecules, proteins, lipids and carbohydrates, combine together to form the interstructure of cell membrane, as we know, according to this fluid mosaic model, which is proposed by Singer and Nicholson in 1972. The first macromolecule we are going to discuss is the lipids. As we know, the lipids constitute nearly 40% of total. These are chemically significant. These lipids are chemically significant. So these lipids are three types. I'm going to see them now. The lipids are combined with phosphate. As we know, they are called phospholipids. Similarly, the lipids combined with the carbohydrates, as we know, they are called glycolipids. The lipids that are combined with the third type of lipid that are not combined but they are form of sterol. For example, cholesterol. The sterol form of lipid that is present in cell membrane is cholesterol. Cholesterol is an important component. We need to remember its clinical significance. We are going to discuss later. So there are three types of lipids in the cell membrane: the phospholipids, the glycolipids, and the sterol form of lipids, that is cholesterol. So first is the phospholipids. This phospholipid, first thing we are going to see is phosphatidylcholine. This is the chemical form, or general form we known as phosphatidylcholine or lecithin. This phosphatidylcholine or lecithin is clinically significant in lungs. This lecithin is an exclusive component of surfactants in lungs. So now remember, this lecithin is an important component in lungs. It's an exclusive component of the surfactants. The surfactant substance reduces surface tension For now, remember, we are going to discuss full details in the respiratory physiology modules. For now, remember, this lecithin is the exclusive component of surfactant in lungs. Second, we are going to discuss in the phospholipid is DPPC. DPPC stands for dipalmitoyl phosphatidyl choline. First, we have seen only phosphatidyl choline, that is lecithin. Next, we are going to see is dipalmitoyl phosphatidyl choline. This is the form of the lecithin. It is also present in the surfactant of lungs. To prevent the collapse of lungs. As we know, the surfactant reduces the surface tension, it prevents the collapse of the lungs. This deficiency of surfactant in infants, clinically we are going to discuss this deficiency of surfactant in infants. They are prone to this disease called HMD, which stands for hyaline membrane disease. So the deficiency of surfactant is more prone or common in infants, which leads to HMD, this hyaline membrane disease, in which there occurs collapse of alveoli. So alveoli is a important structural component of the lungs. So deficiency of the surfactants in the lungs leads to hyaline membrane disease, in which there occurs collapse of the alveoli. So this is the clinical significance of phosphatidyl choline, or more specifically to be dipalmitoyl phosphatidyl choline or lecithin. Next phospholipid we are going to discuss is sphingomyelin. Sphingomyelin. As you can see from this name, there is myelin, so it is involved in myelination of nerve fibers. Very simple, sphingomyelin, myelination of nerve fibers. It's also a minor surfactant in lungs. You have seen the major surfactant in lungs is the dipalmitoyl phosphatidyl choline, or simply phosphatidyl choline. The sphingomyelin is a minor surfactant in lungs. This both lecithin and sphingomyelin, they are used to assess fetal lung maturity. To assess the fetal lung maturity, this both lecithin and sphingomyelin are used. We have a lecithin by sphingomyelin ratio, which is lecithin by sphingomyelin ratio. This L by S ratio must be greater than or equal to 2. To assess fetal lung maturity, we have lecithin by sphingomyelin ratio, which must be greater than or equal to 2, which means fetal lung is mature without any defect. This ratio greater than or equal to 2 indicates the fetal lung is mature without any defect. So these two are the chemical significances of phosphatidylcholine, lecithin and sphingomyelin. The next we are going to discuss is phosphatidylserine. Phosphatidylserine. It's another phospholipid. It's phosphatidylserine, short form PS. And the sphingomyelin, short form to S, as we seen in this ratio, and in this lecithin the short form is L. So this phosphatidylserine short form is PS. So we are going to see how they are significant here. So it's an important fact that the lipid in cell membrane is bilayer, that is arranged in two layers. It's called lipid bilayer. This lipid in the cell membrane is arranged in two layers called lipid bilayer. So we are going to see these two layers. This is the outer layer, this is the inner layer. This is the outer layer, this is the inner layer. On the outer layer we have a lecithin molecule. Well, for lecithin we have a lecithin molecule. On the inner layer we have a phosphatidylserine molecule, like this. In the outer layer we have a lecithin molecule. In this inner layer we have a phosphatidylserine molecule. This is a feature of a normal cell. In a normal cell membrane, this is a feature that the outer layer contains lecithin and the inner layer contains phosphatidylserine. This is called asymmetrical distribution. These two layers of arrangement of lecithin and phosphatidylserine is called asymmetrical distribution, which is a feature of a normal cell. Now if this arrangement is reversed or in any way modified, this is the outer layer, this is the inner layer. Suppose in this outer layer we have found by chemical investigation there is phosphatidylserine instead of what? Lecithin. In this outer layer we have found phosphatidylserine. This is a feature of abnormal cell. In the outer layer lecithin is a feature of a normal cell, but in the outer layer presence of phosphatidylserine is a feature of an abnormal cell. This presence of phosphatidylserine is very important clinically and pathologically, as this leads to a signal called eat me signal. This phosphatidylserine leads to a signal generation called eat me signal, that is, as we have commonly known, a form of programmed cell death, which we commonly know as apoptosis. In the abnormal cell, presence of phosphatidylserine in the outer lipid layer leads to a signal called eat me signal, which means cell destruction signal. It's a form of programmed cell death which is called apoptosis. It's very important pathologically. Features we are going to learn in the pathology subject in second year. For now, remember this phosphatidylserine plays an important role in apoptosis, where it is present in the outer layer, the abnormal cell. This abnormal cell needs to be destroyed. So this apoptosis takes place. It's a form of programmed cell death. So we've seen the clinical significance of phosphatidylcholine or lecithin, sphingomyelin, and phosphatidylserine. Now an important compound here we need to see is annexin 5. Annexin 5. This annexin 5 combined with phosphatidylserine. This PS stands for phosphatidylserine. Annexin 5 combined with phosphatidylserine forms complexes. It forms complexes, and this can be detected. This is a very important information we need to learn, that is annexin 5 combines with phosphatidylserine to form complexes which can be detected. This is called annexin 5 staging. Very important in diagnosis. This annexin 5 staging is important. This combines with phosphatidylserine, which complexes can be detected. In the next phospholipid we are going to see is phosphatidylinositol. This phosphatidylinositol, in short, we are going to write as IP3. This IP3 acts as a second messenger in cellular transmission of signals. This phosphatidylinositol, short form IP3, it acts as a second messenger in cellular transmission of signals. Its major function is to cause influx of calcium ions. This IP3 causes influx of calcium ions, so this is called IP3 DAG, calcium secondary messenger system. So the most important purpose of this phosphatidylinositol is IP3-DAG calcium secondary messenger system. Acts as a second messenger which causes influx. So what is, remember, influx of calcium ions. Next one important phospholipid we are going to discuss is cardiolipin. There are two words here, cardio and lipin. We have already known from the word that it's a lipid which is present in the cardiac muscles, that is exclusively in heart. In heart, cardiolipin is exclusively present. Where exactly? To be precise, in the mitochondria of cardiac cells. In the mitochondria of the cardiac cells, this cardiolipin is abundantly and exclusively present. So what is this clinical significance? So there are autoantibodies. There will be autoantibodies against cardiolipin. Autoantibodies against this cardiolipin are called anti-cardiolipin antibodies. Autoantibodies against cardiolipin are called anti-cardiolipin autoantibodies. Due to infection due to this microorganism Treponema pallidum. This Treponema pallidum, this microorganism, which leads to this common disease syphilis. Treponema pallidum leading to syphilis. The patient with serum with syphilis, the patient affected with syphilis in this serum, there will find positive for anti-cardiolipin antibodies in a test we called BDRL test. It's a test to identify the venereal disease or sexually transmitted diseases. This VDRL test is done to detect syphilis. The patient will come to the OPD with the symptoms. Serum will be collected and tested. With the infection of this microorganism Treponema pallidum, which causes syphilis. In patient with serum with syphilis, it will produce positive for anti-cardiolipin antibodies. The test we do, that is the VDRL test. So this is the clinical significance of cardiolipin. It is exclusively present in the mitochondria of the cardiac cells. It is exclusively in heart. So we have seen couple of phospholipids. First we have seen phosphatidylcholine. Most clinically important form is dipalmitoyl phosphatidylcholine, which acts as a surfactant in the lungs. Next is sphingomyelin. Next we see the phosphatidylserine. Next we see phosphatidylinositol. The next important phospholipid we see in heart is the cardiolipin. Next type of lipid we are going to discuss is glycolipids. Glycolipids are lipids combined with carbos, that is our carbohydrates. The phospholipids are the lipids combined with the phosphate. The glycolipids are lipids combined with carbohydrates. So we are going to discuss one by one. First we are going to discuss these glycolipids are present in two important places in CNS, that is brain, and in GIT, that is gastrointestinal tract. In CNS or brain and in GIT, the commonly present glycolipids are gangliosides and cerebrosides. You must have seen all these compounds in biochemistry. These are important compounds, gangliosides and cerebrosides. These are an important example of glycolipids. So clinically significant gangliosides are GM1 gangliosides or GM1 ganglioside. This GM1 ganglioside it serves as a receptor for entry of Vibrio cholera toxin. Vibrio cholerae toxin. It serves as a receptor for entry of Vibrio cholerae toxin, which leads to diarrhea and in fatal cases can also lead to death if not treated. So these glycolipids importantly present in the central nervous system, the brain, and in the GIT. They are the gangliosides and the cerebrosides. Clinically important, the GM1 gangliosides. It serves as a receptor for entry of Vibrio cholerae toxin, specifically present in the GIT. This Vibrio cholerae toxin leads to diarrhea. We commonly know cholera, and in all fatal cases can lead to death. So the next form of lipid we're going to see is the sterol form, that is cholesterol. This cholesterol is already an unique compound. It's a very important compound in the functioning of cell membrane. This cholesterol first going to see in skin. When sunlight falls on the skin, this cholesterol is converted to 7-dehydrocholesterol in the skin in presence of sunlight. The range is 290 to 315 nanometers of wavelength. This wavelength and falls in skin. This cholesterol is converted to 7-dehydrocholesterol, which after many successive reactions forms our important vitamin D. This is the first important. So this cholesterol is the precursor of vitamin D in our body. In the cell membrane, its function is fluidity. The fluidity of cell membrane is maintained. So what is important? Maintained by cholesterol. It neither increases nor decreases. This we have to remember. It's maintained and it neither increases nor decreases. Commonly asked questions are: what is the role of cholesterol in the fluidity of membrane? Either increases or decreases. It does neither. It maintains within a range. Fluidity of cell membrane is maintained within a range by cholesterol. In the cell membrane, as we see, the model named is fluid mosaic model. So this fluid component is by lipids. If the cell membrane is properly fluid, it will properly function. This proper function of cell membrane, this membrane needs to be properly fluid enough, which is maintained by our lipids, that is cholesterol, the sterol form. Increased in fluidity is good for our health in general, or increased fluidity of cell membrane is good for health. Example, fatty acids, especially the unsaturated fatty acids, they are also called essential fatty acids. AFA stands for fatty acids, also called essential fatty acids. Some examples are linoleic acid, linolenic acid, and arachidonic acid. The examples are linoleic acid, linolenic acid, and arachidonic acid. These are examples of unsaturated fatty acids, also called essential fatty acids. Why they are essential fatty acids? Because these fatty acids increases the fluidity of membrane, which is in general good for health. So that's why these unsaturated fatty acids are also called essential fatty acids. In fish, it's rich in omega-3 fatty acids. So omega-3 fatty acids are rich in the fish. These are also examples of unsaturated fatty acids. Some unsaturated fatty acids are essential fatty acids. So omega-3 fatty acids are essential fatty acids. So we have seen this phenomenon, increase in fluidity. Now we are going to see decrease in fluidity. This decrease in fluidity is caused by saturated fatty acids. The saturated fatty acids causes decrease in fluidity. This decrease in fluidity in excess causes static cell membrane, that is not enough fluidity. We've seen this fluidity is important for functioning of the cell membrane. The saturated fatty acids, which decreases fluidity in excess, causes static cell membrane. The cell membrane is too static, not much fluidity is there, which is in general bad for health. So we're going to see example which are the saturated fatty acids. Stearic acid and palmitic acid. The stearic acid and palmitic acid are the saturated fatty acids which decreases fluidity. In general, it is bad for health. These fatty acids are called trans fats. The saturated fatty acids are also called trans fats. We have most commonly seen this term trans fats, most common in junk foods. That is why it is always advised to maximally minimize our consumption of junk food, as they are bad for health. They causes static cell membrane. No, there is not enough fluidity. The stearic and palmitic acid are examples of saturated fatty acids. These are trans fats in the junk food. So this cholesterol does maintenance of fluidity. You have seen and understood why it neither decreases nor increases the fluidity, it maintains the fluidity. So this is why it's also called fluidity buffer. This maintenance of fluidity is within a range, so that's why it's called, in this important term, fluidity buffer to cholesterol. This cholesterol is known as fluidity buffer. So with this we have seen the important forms of lipids in our cell membrane: the phospholipids, the glycolipids, and the sterol form, that is cholesterol. Now we are going to discuss is the next important component, proteins. Proteins are the major for about 55% of total. Proteins are the major form, constituting about 55% of total. So initially we have seen this fluid mosaic model. This fluid term is for lipids. The remaining mosaic term is for proteins. These proteins are also called mosaics. So fluid mosaic model is based on the lipids and the proteins. These proteins are also called the mosaics. So we are going to see the types of proteins. There are, more importantly, 3 types of proteins. 3 types of proteins we are going to see. So this is the outer layer, this is the inner layer, and we're going to see this one protein. This second type of protein, and this third type of protein. So there are 3 types of proteins here. Let me discuss them one by one. This first one is called transmembrane proteins or integral membrane proteins. Integral membrane proteins. So called transmembrane proteins or integral membrane proteins. Why? Because they span across the cell membrane. This is the outer layer, this is the inner layer. They span across the membrane, so this is called transmembrane protein or integral membrane proteins. The next type we are going to discuss is this one. This is called peripheral membrane protein. On cell surface. So on this cell surface we have this peripheral membrane protein. The second type of protein you see. And this third type of protein you see is which is lipid anchored protein. It is attached to lipid. This protein is attached to lipid So this is called lipid anchored protein. So these three types of proteins we have seen. The transmembrane proteins or integral membrane proteins. This protein spans across the membrane. The second type protein is the peripheral membrane protein, present on the cell surface. And this is the third type protein attached to lipid anchored protein. So the first we are going to see transmembrane protein. Transmembrane are also called integral protein membrane. We have seen. Most important example are the receptors. The receptors are the most important examples of the transmembrane proteins. So we see these are two layers. One, so these are two layers. We are going to span this protein 1, 2, 3, 4, 5, 6, 7 times. Sorry, excuse me. This is going out, I can't understand. So we draw this 7 times. So this receptor, it spans the membrane 7 times, so called serpentine receptors. This membrane protein spans the membrane 7 times, called serpentine receptors. Important example of the serpentine receptor is this GPCR. It is G protein coupled receptors. These GPCR or G protein coupled receptors are examples of these serpentine receptors, which are transmembrane proteins. This GPCR example is insulin receptors. So these insulin receptors are examples of GPCRs. Another example of this transmembrane proteins are the pumps. You know, various pumps exist across and inside these cell membranes and the cells. Example, sodium potassium pump. Very important pump, sodium potassium pump. These pumps are example the transmembrane proteins. Next example are the ion channels. As these pumps, these ion channels are also the transmembrane proteins. For example, this chloride ion channel. This chloride ion channel is an example of transmembrane protein. This chloride ion channel is clinically significant, as we are going to discuss here, is CFTR. It is cystic fibrosis transmembrane regulator. The cystic fibrosis transmembrane regulator or CFTR. This is important in regulating opening of chloride ion channels. The CFTR, cystic fibrosis transmembrane regulator, it regulates the opening of these chloride ion channels. Remember, chloride ion channels. So if any gene of CFTR undergoes mutation, leads to cystic fibrosis disorder. So gene of CFTR undergoing mutation leads to cystic fibrosis disorder, very commonly seen disorder in the, especially in the lungs of individuals. So the important, clinical important, very important, CFTR is controlling chloride ion channels, and the mutation causes cystic fibrosis disorder. The next type of protein I'm going to discuss is peripheral proteins. First we have seen the transmembrane proteins. The examples are receptors, ion channels, pumps. Next one is the peripheral proteins. These peripheral proteins are generally supportive in nature. These peripheral proteins are generally supportive in nature. They are most important at two places. First one is this all important RBC. Second one is skeletal muscle. So these peripheral proteins are generally supportive in nature, present in RBC and skeletal muscle. They support the structure of RBC and skeletal muscle. So the skeletal muscle and this RBC structure are supported by these peripheral proteins. Now we are going to see what are these peripheral proteins that are important in RBC and skeletal muscles. In RBC we have got, first is spectrin protein and ankyrin. The spectrin and ankyrin proteins are the two important peripheral proteins important for the supporting structure of the RBC. As they are important, I'm going to see one by one what happens in their respective disorders. In spectrin, if undergoing mutation, leads to hereditary elliptocytosis. Hereditary elliptocytosis. We commonly know that the RBC is oval shaped or spindle shaped, but the spectrin protein, when undergoing mutation, it leads to this elliptical shaped RBCs. This is called hereditary elliptocytosis. And next, this ankyrin protein, if undergoing mutation, it leads to hereditary spherocytosis. The RBC cells become spherical in shape. Here the RBCs become elliptoid in shape. So what's the problem going forward? So this disorder in shapes, this leads to, as we know, the spleen. In the spleen, this is the graveyard of RBC. After completing the life cycle of 128 days, the RBCs are destroyed in spleen. in the spleen, as the shapes are altered, the disorder in shapes, in spleen there occurs extensive destruction by macrophages. There occurs extensive destruction by macrophages. This leads to commonly hemolytic anemia. So this disorder in the shapes in spleen leads to extensive destruction by macrophages. The macrophages are the destroying molecules. It leads to hemolytic anemia. So now we have seen the two proteins, pectin and ankyrin, and their hereditary disorders. What happens if they undergo mutation? Now we are going to see proteins in the skeletal muscle. First we are going to see this important dystrophin. This dystrophin is important, very important in skeletal muscle. It acts like an anchor. It acts as an anchor. Its mutation of dystrophin can lead to this condition called Duchenne muscular dystrophy. As the name suggests, dystrophin, its mutation leads to Duchenne muscular dystrophy. Duchenne muscular dystrophy features are muscle weakness. Obviously, as an important protein, its mutation leads to muscle weakness, especially in the children. The affected child does not stand up immediately. Child is affected with this condition, it does not stand up immediately. This is an important feature. When we ask the child to stand up on its own, it does not stand up immediately. It uses its own body to climb and stand. It uses its own body to climb and stand. This is commonly called Gover's sign. This, when we test the patients with this Duchenne muscular dystrophy, these two important features are seen. It does not stand up immediately, and it uses its own body to climb and stand. This sign is called Gover's sign. Also, in this mutation of dystrophin, the diaphragm is an important respiratory muscle. This diaphragm is affected. So this leads to respiratory muscle paralysis. The diaphragm is affected, causes respiratory muscle paralysis. In fatal conditions, can lead to death. An important muscles and respiration, if it undergoes paralysis in fetal conditions, can lead to death. So in the skeletal muscle, we have seen this important protein dystrophin. Its mutation can lead to this Gover's sign and affecting the diaphragm muscle. to see these two peripheral proteins in RBC and skeletal muscle. The third type we are going to see is lipid anchored protein. Proteins are anchored to the lipid. Common example, as we have seen before, is this IP3. So IP3 molecule is an important lipid anchored protein called GPI proteins. GPI proteins. This PI, as we know, stands for phosphatidyl inositol. Phosphatidyl inositol is IP3 called GPI proteins. So we are going to see what are these lipid anchored proteins. We are going to see on the RBC molecule. This is a molecule of CD55 receptor and CD59 receptors. This is RBC molecule. You have C55 receptor and CD59 receptor. The functions of these two receptors are, when the proteins are exposed, it prevents hemolysis of RBCs. To prevent this hemolysis of RBCs, the exposition of these proteins are important. What are these proteins? The CD55 and CD59 present on the RBC molecule. So obviously, when absence of these proteins, RBC undergoes hemolysis. As they are preventing the hemolysis of RBC, their absence causes hemolysis. It happens predominantly during night times. This is called an important condition, paroxysmal. It happens during the night. Nocturnal hemoglobin urea. Paroxysmal nocturnal hemoglobin urea, PNH. This paroxysmal nocturnal hemoglobin urea, or this PNH condition, are due to absence of these two lipid anchored protein, CD59 and 55 receptors. This is important as when these proteins are exposed, it prevents hemolysis of RBCs, and their absence causes PNH, paroxysmal nocturnal hemoglobin urea. So we will discuss the two important macromolecules. The first is the lipids. We have discussed various subtypes of lipids. Then we have discussed the second macromolecule, the most important, that is protein. We've seen three types of protein: lipid anchored protein, peripheral proteins, and transmembrane proteins. I've seen some of their examples, the clinical importance, and what happens when their gene is mutated or they are absent. The third macromolecule we are going to discuss is membrane carbohydrates. Let's consider the least important, about 5% of total. Carbohydrates are total 5% of the total. So how they're important we are going to see again in the RBC. For blood group detection, there exist these sugars. The presence of this sugar lead to different blood groups. So we're going to see the blood groups here. First is A blood group. How this A blood group is determined? On the RBC we have this molecule called NAG, which stands for N-acetyl galactosamine. This is not N-acetyl glucosamine, this is N-acetyl galactosamine. This presence of N-acetyl galactosamine on the RBC determines the A blood group. You can remember from this NAG, this N-acetyl leads to A blood group. Presence of this sugar on the RBC confers A blood group. Next we are going to see B blood group. In this B blood group, in the RBC, there is presence of this sugar. It is only G, which stands for galactose. Presence of this galactose sugar on the RBC determines the confers B blood group. You can remember then there is no A here, only galactose. So galactose confers B blood group. There is A here, N-acetyl galactosamine, A blood group. Presence of galactose, B blood group. The third I'm going to see is O blood group. This O blood group, the RBCs have no sugar, no sugars on RBCs. Then circle this O, this no sugar confers O blood group. N-acetyl galactosamine A blood group, galactose no A, B blood group, and no sugars, neither NAG nor G, nothing are present, O blood group. Next is both NAG, N-acetyl galactosamine, and galactose are present. This is our AB blood group. On the RBC molecule, both these molecules are present and both are equally expressed, leading to AB blood group. So these are the importance of the membrane carbohydrates. This is generally for blood group detection. AB and O blood groups, where we have seen the different sugars that are involved in this. We have completely discussed the three important macromolecules. It is the lipids, proteins, and carbohydrates. Some facts we are going to discuss now. The percentage of proteins is 55%. Percentage of these lipids is 40%. In ideal general conditions, these proteins are generally greater in concentration than the lipids, 55% and 40% respectively. We are going to see the highest protein concentration, it is 70% in inner mitochondrial membrane. In the inner mitochondrial membrane, there is highest protein concentration of any other structure, contains 70%. As exception to this generalized rule, this exception is lipids are 80% and proteins are 20%. It is lipid is greater than protein, where we see this in myelin of nerve cell membrane. In myelin of this nerve cell membrane, we see this exception of lipid being greater than protein concentration, lipid being most 80% and protein restricted to 20%, where in myelin of nerve cell membrane. In inner mitochondrial membrane, we see the highest protein concentration, 70% in the inner mitochondrial membrane. Now we are going to see some facts about repair of cell membrane. This repair of cell membrane occurs by resealing of lipid bilayer. This repair of cell membrane occurs by resealing of lipid bilayer. This for minor injury. This for small gaps. For minor injury to the cell or creation of the small gaps, the repair of cell membrane occurs by resealing of the lipid bilayer. For major injury, that is creation of the big gaps, we have repair by calcium dependent processes. For major injury to the cell membrane, occurrence of big gaps, they occurs repair by calcium dependent processes. Processes are dependent on this calcium ion. For minor injury or creation of small gaps, there occurs resealing by lipid bilayer. So this concludes today's video. We have seen the three important molecules, macromolecules, the lipids, proteins and carbohydrates, and their subtypes, how they are clinically important, the clinical manifestations we have seen, how their mutation or absence affects our general metabolism. So please do revise them, guys. Thanks for watching this video. I will catch you in the next one. Good luck.